Today we're going to be talking about RNA and the process of transcription. If you think about what the word transcription means, or a person who would transcribe something, you'll probably think about scribes in ancient Egypt, who were people that were trained to write things down, because not everyone knew how to read and write. And that's exactly the same thing that we're going to be talking about here. The DNA needs to stay inside the nucleus. It can't leave, but we're going to need copies of the DNA to go out to the cytoplasm. So RNA will be transcribed or copied from the DNA before that information can actually be used in the next step of translation. So in these notes, we want to talk about the different forms of RNA and know why there's different forms of RNA. And then we'll go through the actual process of transcription and talk about how and when that RNA would be formed. This is a URL link for you that you can watch if you want to just see a quick overview of the process. So as you guys know from your outlines, RNA has some differences from DNA. There's three major differences that you should know. The first and most obvious one is that RNA is a single strand while DNA is a double strand. Only one side of the DNA strand holds the code for the gene, so only one side of the DNA strand will be copied during transcription. The other two differences are more specific, and they're changes in the actual nucleotide. First off, instead of having A, T, G, and C, RNA has A, U, G, and C. So instead of having thymine, we have the base called uracil. Uracil just simply takes the place of thymine, and anywhere you would originally have wanted to put a T, now you will put a U. And then the other difference with the nucleotide is that instead of having a deoxyribose sugar, like the DNA, we have just a ribose sugar. So there's the difference between D and R. It's not a deoxyribose sugar, it's just a ribose sugar. There's many different forms of RNA, but the three most common that we'll talk about the most in our work here are mRNA, tRNA, and rRNA. So all RNA, when it first forms in transcription, looks a lot like mRNA, and then there's enzymes in the cell that can modify the RNA to make it look, to turn it into its other forms or make it look different. So starting off with mRNA, mRNA is the messenger RNA. This is the actual copy of the gene. The gene can be between, about 100 and 1,000 base pairs long. This is what will go out of the nucleus and go dock with the ribosome for translation. tRNA is also going to be really important in that translation process. It is called transfer RNA because it transfers amino acid. And you can see that amino acid right here at the end of the tRNA molecule. The tRNA, you can see, still has a little bit of that same backbone that we see over here in the mRNA. We see that folded up and greatly modified in the tRNA, and it does still have some base pairs. These base pairs are going to be known as the anticodon, whereas the base pairs over here in the mRNA are going to be known as the codon. So we'll write that down. Codon, anticodon. And this terminology is going to become more important when we talk about translation, because when the actual protein is being formed, the codon from the mRNA will match up with the anticodon from the tRNA. And when they match together, it's going to work just like the DNA did. Instead of having T's, remember we have U's, but it'll go A to U and G to C so that we can interpret this code and build a protein. Okay, so we have the mRNA and we have the tRNA. The third most common form of RNA is called rRNA, or ribosomal RNA. And you can see from the picture that this ribosomal RNA is embedded within the ribosome. It's very highly modified, and it just helps to make the ribosome work so that the ribosome can make proteins. So all three of these forms of RNA will be formed in the process of transcription, but they'll be used later to perform the process of translation. Let me say that one more time. All three of these forms of RNA will be formed through the process of transcription. Transcription is the copying of the DNA into RNA. But these three forms of RNA will be used later in the process of translation to actually make the protein. So we'll hear about these again throughout the rest of the chapter. Now, 10 or 15 years ago, we would have just learned about those 3 forms of RNA, but now we actually know that there's many other types of RNA. What we used to believe was that in our genes, or in our chromosomes, where we could find our genes, and that the space in between the genes was just junk. It was just filler. So if you have your whole DNA sequence, we believed that this area right here could be a gene, and this area right here could be a gene, but that the space in between was just junk, or basically just a placeholder. What we found is that that term junk is very inappropriate. In fact, we don't use it anymore. And that those space holders, that extra space in between our genes, is actually most of the time new forms of RNA. Some of these new forms of RNA have pretty amazing functions. So let's talk about some of those functions. These regions of the DNA can be copied in transcription, but instead of being turned into mRNA, or tRNA, or rRNA, they can be turned into snRNA, or the different types of RNA found in a complex called RNAi. snRNA is another type of RNA that is important in maintaining your telomeres. Now, your telomeres, this might be a new vocab word for you guys. Your telomeres are kind of like the aglets at the end of your shoelaces. If you don't know what an aglet is that little piece of plastic right at the end of your shoelace that keeps your shoelace from fraying. A telomere is like an aglet, but on your DNA. So every time your DNA copies in replication, every time your cell goes through mitosis, it can fray the ends of the strands of the DNA. So you actually have these regions. They're not plastic, of course. They're made up of base pairs, but you have these regions at the end of the chromosome that are there to protect the end of the chromosome. And as we age, these telomeres do wear down the same way that an aglet on a shoelace could wear down. So what we found is there's actually a special type of RNA that's coded for in what we used to call these junk regions, this snRNA, which actually codes for enzymes that can cut and slice and maintain these telomeres. So this is definitely something we'd have to look at if we want to increase lifespan in humans, if we want to look into the aging process, because younger people have well-maintained telomeres, and as we age, they start to break down. So this snRNA might be some kind of clue into studying aging. Another new type of RNA is called RNAi. RNAi, as you can see here in the list, is actually 4 different types of RNA. siRNA, miRNA, shRNA, dsRNA. It's a whole bunch of RNAs that work together. And this is really very complicated, so I'm just going to try to explain this in as simple forms as I can. It's something that's called gene silencing. And basically what this means is these 4 different types of RNA can work together to turn genes on and off. It was actually studied in plants first. They were studying plants and expecting the plants to come out a certain color, a purple color, and all of a sudden all these plants were coming out white. All the petals in these plants were coming out white, and they couldn't figure out why. And so they actually, in researching these begonia plants, finally figured out that there was a complex of RNAs, these RNAi complexes, that were actually turning the color genes off so that the plants didn't have the color that they were supposed to have. Now again, this is something that we might use in medicine or use in future research, because if we had the ability to turn a gene off, to silence a gene, or to turn a gene back on that wasn't working correctly, this could really influence things like medicine or medical treatments. We are not going to focus too much on these new types of RNA, but I want you to be aware that they're out there, and most importantly, I want you to understand that this term junk DNA is no longer appropriate, that there are not just blank fillers in between all of our genes, but that most all of the DNA does have some purpose. The overall process of transcription is actually much simpler than what we just did with replication, because it is just a shorter, quicker process, and there's only one enzyme that you have to learn. The enzyme is polymerase, but you'll notice it says RNA polymerase, so it's going to work very similarly to the DNA polymerase that we saw earlier with our replication. Basically, when we do transcription, we're not copying all of the DNA. We're only going to copy one gene. And so we need to focus in on that one coding region, or that one area of the gene that we want to copy. And in front of that gene, we have what is called a promoter. it's just a signal to the RNA polymerase that, hey, this is where the gene's about to start. And then at the end of that gene, we have a termination site. Again, just a signal to the RNA polymerase that, hey, this is where we're going to stop making the copy. So RNA polymerase is able to bind to the promoter. It copies the coding region, and then it lets go at the termination site. It's a very simple process. It only copies one gene at a time, and it's only copying one side of the DNA strand. That RNA transcript is going to initially look like mRNA. It'll go out into the nucleus, out of the nucleus if it's mRNA, and go out to the ribosomes for translation, or it could be modified into one of the other types of RNA like tRNA or rRNA. When this transcription is happening, remember that the coding rules still apply, so the basics of it still look pretty much like replication. As the RNA polymerase, after it finds the promoter and it's traveling along the coding region, it's just matching A's to U's, G's to C's, and that will just continue until it reaches the termination signal and that everything lets go and that transcript can leave the nucleus and go out to the cytoplasm. Since the DNA cannot leave the nucleus, this is a really important step so we can get a copy of that gene out to the ribosomes so we can do transcription. So hopefully you're seeing this connection between the DNA and the ribosome. mRNA is just this little shuttle bringing the code from one place to the other since the DNA is not allowed to leave the nucleus. Remember that when transcription initially happens, all of the transcription typically looks like mRNA, right? We copy the coding region, just one side of the DNA. We make our single-stranded RNA that has ribose sugars and A's, U's, G's, and C's in any pattern that we want. Just copying that gene. But modification can happen. This can be modified to be turned into tRNA. It can be modified to be turned into rRNA or the snRNA or any of those RNAis that we've already talked about. Finally, it's important to remember that not every piece of the DNA is going to be copied and then turned into protein. Some of the DNA are those spacers. Remember, we don't call it junk anymore. That's a bad term. But some of the DNA is not coding DNA. It's not something that's going to eventually be turned into proteins. It might be something that will be turned into one of those special new types of RNA. Instead of saying junk, now what we do is we refer to the DNA as either an exon or an intron. Exons will be expressed, so that's easy to remember. Exons will be expressed as DNA, and the introns will not be expressed. The introns will stay in the nucleus. So if you look at the picture here, you have your DNA, so you have your coding regions and your non-coding regions. Coding regions and your non-coding regions. All of it is copied, so when we have transcription, the whole thing is copied, but then there will be a modification step where the introns will be spliced or cut out, and only the exons will be actually sent out of the nucleus to be transcribed. So again, to be translated. So again, this connection between transcription and translation. We transcribe the DNA into RNA, and then once the RNA has been modified to only contain exons, this final piece of RNA will go out of the nucleus to the ribosomes, and that's where translation will occur. We transcribe the DNA into RNA. It has to go through this modification process so that it only contains the exons, and then those exons will be expressed into proteins in that next process of translation.